Hello and welcome to today's seminar with Alzecure Pharma. Before I hand over the word to the CEO, Märta Jönsson, I will give some practical information here. If you want to send in a question for the Q&A session, you can use the form to the right to submit your question, and we will then take it during the Q&A. And with that said, I hand over the word to you, Martin.
Thank you so much, Ludwig. Welcome everybody to today's seminar. And the seminar we will have is on TrkA-NAM, our pain project, which is a novel non-opioid treatment of severe pain conditions. And today's presenters will be Märta Segerdahl, who is our Chief Medical Officer at Alzecure Pharma. And Märta, she is an MD, PhD. She has done research on Karolinska Institute and has a long background in several global companies such as Lundbeck and AstraZeneca, as well as Grünenthal. And then we have our head of discovery and research, Pontus Forsell, who is a PhD, who has done research and also a PhD in medical biochemistry and biophysics from Karolinska Institute. And then you have me, Martin Jönsson. I'm the CEO of Alzecure Pharma. And the agenda for today is that we will have a brief introduction to Alzecure as a company, what we are focusing on our business model. After that, we will go in and talk about the unmet medical need and the opportunities for TrkA-NAM in pain treatment. And thereafter, we will go into TrkA-NAM and look at it as a next generation non-opioid analgesic. And this presentation will be done by Pontus Forsell. And then we will go into Q&A session where you have the opportunity to send in questions. And then we will have concluding remarks. Going in and talking about Alzecure very briefly. We are a company who are focusing on Alzheimer's disease and pain. And we are a spin-out out of AstraZeneca. And we were founded in 2012 and became a commercial company in 2016 with funding from the Swedish Alzheimer's Fund. And we are based at Karolinska Institute. And we are developing small molecule candidates, first-in-class properties and best-in-class properties. And we have one project called Alzstatin, which we developed to be a preventive and disease-modifying treatment against Alzheimer's. And then we have New Restore, which is a novel symptomatic treatment of cognitive disorders to improve learning and memory capabilities, where we are focusing on Alzheimer's disease, but also see opportunities in several other indications, such as depression. And then we also have a platform for pain projects. And here we are also working with neurotrophins, which we then are blocking off to treat pain conditions. And as a company, we are listed at Nasdaq First North Premier Growth Market here in Stockholm since 2018. And our business model is that we are a research and development company. We research and develop projects into early clinical phase and then to out-license them. And with out-licensing, our goal is to use this to drive our other projects forward. And our pipeline of small molecule programs looks like the following. We have New Restore, which is a positive allosteric modulator to treat Alzheimer's and other cognitive disorders. And here we have a positive phase 1. And now we are preparing this project for a trial in Alzheimer's patients. And here we have gotten a grant from the European Innovation Council, which we got last year. And we are now preparing to start a trial in early Alzheimer's patients this year. Then our project Alzstatin, here we last year selected a clinical candidate, which we now are preparing for a clinical phase. And this is a treatment to prevent that individuals are developing Alzheimer's, but also to treat individuals who have Alzheimer's. And then going into our pain project, we have ACD440, which is a TRPV1 antagonist, where we focus on neuropathic pain and also are working with orphan. And here our orphan indication is erythromelalgia. And we have positive data in chronic patients with peripheral neuropathic pain, showing very positive safety, tolerability, and pain reduction. We diminished pain with around 50% in patients with chronic peripheral neuropathic pain, which were temperature-induced. And we also have received an orphan designation from the FDA for the indication erythromelalgia. And we this year, earlier, a couple of months ago, also got an orphan designation from the European Medical Agency. So this project, we are now preparing for the next phase. And from the FDA, we have also for this indication, pain in erythromelalgia, received positive feedback on a potential pivotal trial from the FDA. And then we come to the project that we will talk more about in detail today, and that is ACD137, which is a negative allosteric modulator where we focus on osteoarthritis. And here we have selected a CD, which we have developed in-house, and we also have several other molecules in this series. And lately, we have published data on TrkA-NAM ACD137, which we are welcome to share with you and that you can download with this QR code. And when it comes to the market that we are targeting with regard to TrkA-NAM ACD137, this is osteoarthritis. And osteoarthritis is a growing market and a very big market. There are today more than 500 million who are suffering from osteoarthritis. The market is worth around $10 billion and growing 5 to 9% annually, and is expected to do so for the coming 10 years. And we are building TrkA-NAM on science, which are well validated both preclinically and clinically. And this is a project that we now look forward to take into the next phase. And to talk about the opportunities for TrkA-NAM with regard to different indications, we will have Märta Segerdahl, who is our Chief Medical Officer, presenting this to you. Märta, please.
Thank you, Martin. So I'm happy to present this to you, a little bit of information on the indications we are considering there's probably many more, but what we are considering right now and where we have non-clinical data. And also I'll tell you quite a bit on the major unmet medical needs in these areas. So when it comes to osteoarthritis pain, you've all heard of it, and you know that you can buy ibuprofen or diclofenac or naproxen to treat your pain, but there's still a huge unmet medical need. Pain starts mostly when you start for physical activities or if you've been too physical in your activities. And later in the disease, you may also have pain during rest and during night. And pain at rest is more common in hip OA. Of course, it's not only knees that can have osteoarthritis, it's also other joints in your body. And swelling and edema is also a problem for these patients. So osteoarthritis, as Martin mentioned, there's more than 1.5 billion people worldwide, according to the WHO Global Report on Disease. And in the US, it's definitely more than 14 million adults that are affected. Symptomatic OA is about in 10% of men and in 13% of women above 60. Not only the knee, as I mentioned, can be affected, but also other joints, hip, spine, hands. So there's a number of different sub-indications in this group as well. And the older you get, the higher the risk is that you actually develop osteoarthritis. Of course, you can have joint replacements, and quite a few people do, but many want to wait for as long as possible. And in some areas, it's not really an option. You can have to wait for several years before you actually qualify for surgery. There's a number of current therapies. I'll tell you a little bit more about those, and also there is a risk for side effects in older individuals. So the higher the risk is that you have osteoarthritis, the higher also is the risk of having side effects with current therapies. So what's in people's pharma cabinets at home now? Of course, exercise is core treatment, both to reduce stiffness and to reduce pain, and very often it's the last resort for patients to actually exercise. Weight reduction, because you have an increased risk of OA if you are heavily overweight. So pharmacological treatments should always be combined with exercise. High-level recommendations actually only for oral non-steroidal drugs like celecoxib, for instance, or ibuprofen or naproxen. High-level evidence is also available for intra-articular injections of corticosteroids, but everyone doesn't want to do that, and you don't repeat it too many times. Other drugs actually many have the indication, but it's a weak level of evidence. The effect size is not very large. Topical treatments are not recommended, nor are strong opioids, though quite a few patients actually receive them. So what are the side effects? What are the downsides with the current treatments? Because if current treatments were perfect, there would be no need for a good treatment, for a new treatment. So a lot of people actually have non-steroidals give them gastrointestinal problems like gastric ulcer, gastritis, diarrhea, nausea. It's about 1/4 of patients, and they actually stop taking the drug due to this. Another 1/4 of patients stop taking them because they don't work at all. In elderly patients, they're extremely much more sensitive to kidney problems. So a large proportion of patients, older patients admitted to hospital, are so because of drug side effects due to non-steroidals. And this is quite unknown, but it's actually a very costly problem for healthcare. Paracetamol has very few side effects, but also very low efficacy. Opioids, patients get constipated, nauseated, they get dizzy, they fall over, etc. And they have an impaired cognition and probably forget their doctor's appointments. So what happened to the NGF antibody story? Because like 15 years ago, monoclonal anti-NGF antibodies were developed, and in phase 3 of clinical development for OA pain and for neuropathic pain and also for cancer-related pain. The effect was absolutely stunning, and it was dose-related. But during the development of Pfizer's, which was a frontrunner of all these projects, the tanezumab, they had some patients developing something called rapidly progressing osteoarthritis. And it could be, that was quite unexpected, and it was mostly in combination with non-steroidals, and non-steroidals were then a rescue treatment. So if the drugs didn't work, they took non-steroidals. So this rapidly progressing OA was dose-dependent, so the lower risk, the lower efficacy. And thus, as a result, anti-NGF antibodies never reached the market because agencies considered that the benefit did not overweigh the risks of this drug. However, they've now been applied in pain management for cats and dogs. So the anti-NGF antibodies have validated our target as a relevant pain treatment, as in ACD137. And we can then, as we will tell you later on, we can avoid the adverse effects seen by the antibodies. So how does then ACD137 differ from NGF antibodies? Of course, that's key information because the problem today is largely safety. So the NGF antibodies, they act, it's released, NGF is released at the site of an injury. If an injury, whether it's peripheral injury or it's an inflammation in the joint, then the TrkA is a receptor for this nerve growth factor, NGF. And it's located on the neuron, the sensory fibers that make you feel pain. And there's also receptors on the immune cells. So you get swelling and you get pain. Now, NGF leads to this inflammation and pain, and the anti-NGF antibodies inhibits this. But they also inhibit some other pathways in the pathway from NGF to the general inflammation, etc. So what happens is that the anti-NGF antibodies are very unselective. So we say that ACD137 is a TrkA receptor negative allosteric modulator. So the NGF antibodies, they stop the NGF, which are very important also for nerve growth, so for reconstitution of injured. They give a pain signal, but they're also important for the restitution of the tissues. But they also take, they take the pain away through the TrkA mechanism. Our drug doesn't take the NGF sort of the inhibitory effects. It only treats the pain part, which is the TrkA. So we avoid all the side effects that were seen with the antibodies that we know now are related to this so-called P75 receptor. So ACD137 can treat pain and stiffness. They can treat pain signaling, and that will improve mobility and wellness, because quality of life is also a very, very important piece of the puzzle for these very many patients. So I will go further into another indication, which we think is very timely, very important, and clearly underserved. And it's a type of peripheral neuropathic pain that is induced by chemotherapy, so cancer treatments. This is a growing patient group. So in 2023, the 5-year survival of cancer was 85% or more in 25 countries in Europe, North America, Oceania. In the US, the 5-year relative survival rose from 75% in the 70s to 90% in 2016. And now the survival rates for the most common and for the most aggressive types of cancer, survival rates are between 99 and 100%, which is quite impressive. That's not what we learned 20 years ago. And I think that's also very much what's in the back of our minds when we think of cancer. So a very important reason for this is the screening programs that have been implemented, at least in the Western world, as well as also improved treatment regimens. Considering the increasing yearly incidence, not the least breast cancer, prostate cancer, colon cancer, and lung cancers, which are the 4 most common kinds, the number of individuals is still increasing because there's more patients who have cancer. Many are caught in screening programs, and many more survive more than 5 years. That means that there's a growing group of people who suffer or can suffer from chemotherapy-induced neuropathic pain. Actually, this group is growing so fast, so there's no really good epidemiological data of how many they are. We just know how many are diagnosed and how many are treated per patient group. But those are longitudinal studies, and data is actually developing over time. Pain after irradiation treatment for cancer is high. In ENT cancers, so ear, nose, and throat, is about 38%, and in general, it's between 30 and 40% of colon and breast cancer are the other 2 indications where radiation is a common treatment. When it comes to chemotherapy-induced neuropathic pain, overall, it does differ between the different chemotherapeutic agents and the type of malignancy. So the intensity of this pain, of the chemotherapy-induced neuropathic pain, often sets the dose limit for chemotherapy. If patients have too much pain, you have to stop dosing and wait until it wanes a bit. So if we can reduce this pain in the acute situation, we could actually optimize the chemotherapy and thereby increase effects further. So chemotherapy-induced pain is very intense in the acute phase. It often attenuates over time, but remaining provoked symptoms can last for very many years in combination with the loss of sensation. Patients can say that it's like they're walking on pins and needles, or walking on Lego pieces if you have them at home, or on coal, depending on how they are. But this is the typical mix. They require steady shoes because they have a loss of sensitivity, and in combination with pins and needles and provoked by cold, even at normal ambient temperature. A typical patient looks like this because patients come and they draw a pain drawing. And what they demonstrate is something that we call a sock. So this is where the pain is, sock and glove distribution. That's very typical. And when the doctor examines the patients, they can say that, okay, you have a loss of sensation here, and that's where we have all these crosses back and forth with the lines that are perpendicular and the cross. And then most of them also have so-called hyperphenomena. So that means that you can elicit pain with cold or with pinpricks, but especially cold in these patients. In the summer, they can sleep with their warm socks on, just to be able to sleep and not be in pain. So what is the current standard of treatment? Same treatments as for other peripheral neuropathic pains is actually used. So it's serotonin, norepinephrine reuptake inhibitors, so duloxetine, gabapentinoids, tricyclic antidepressants, and they have a very poor efficacy, less than in other neuropathic pains. It's not good in the other types, but it's even worse here. It's like 1 to 12 to 15 that you have to treat. So treating 12 to 15 patients to have one with sufficient analgesia is not a very good treatment result. Many patients just need to have better coping strategies, which is tough. There's few studies actually dedicated in chemotherapy-induced neuropathic pain on clinicaltrials.gov. So one study funded by the NIH demonstrated a significant reduction of pain versus placebo. But two ongoing studies are one now 1.7 channel and one calcium channel blocker. We don't know what happens with those. They still have another year to go. And there's one completely negative study with pregabalin. One reason that we don't know more is probably because the grading systems that is used in oncology questionnaires, they don't even ask about pain. They ask about function. Can you handle yourself at home? Can you cook? Can you hold a mug? Can you do your toiletry as you should, etc. So this is not really measuring the problem. But one study actually looked more into this, and they looked at patients who had been treated for colorectal cancer. And it turned out that this was after several years, 9% of the survivors had painful neuropathic pain, and 9% of all those patients who have been treated for cancer, that's quite a few, and non-painful chemotherapy-induced neuropathic pain was reported by 22%, and those 22% also include those with evoked pain, so with stimulation-induced pain. Time since diagnosis was related to chemotherapy-induced pain, and it can actually start a couple of years after end of treatment. Higher disease stage, so higher total dose of chemotherapy is, of course, important and related. And survivors with painful chemotherapy-induced neuropathic pain, they report a much worse global quality of life. So it must be painful chemotherapy-induced neuropathic pain must be distinguished from non-painful patients, as they say only painful patients were related to worse quality of life. I think it's probably a little bit of modification here, because I think they missed a few in the 22% that were actually suffered from stimulation evoked pain. I will not tell you more about this, just a list just saying that there's quite a few, almost all of our most common chemotherapeutic agents, they actually cause neuropathy. It's just very well known, and it's how the symptoms of neuropathy is what drives your maximum dose. So the same as with osteoarthritis, and that my colleague Pontus Forsell will tell you quite soon as well, is that anti-F antibodies, they've shown promise in alleviating pain, also in chemotherapy-induced neuropathic pain. But they were halted because of the side effects that were found in the Pfizer program. And it's not because of Pfizer, because it was there's a target-related problem with the NGF antibodies being unselective and not being only effective on the TrkA, which is the part of the NGF downstream activity that is related directly to pain. So this one I already showed you, just for remembering. And so in summary, OA pain affects more than 500 million people worldwide, and it increases because the aging population increases. Current therapies are not very efficacious. They have limited tolerability. Chemotherapy-induced neuropathic pain is growing. Very few have an effective treatment. 9 to 10% of the patients develop chronic neuropathic pain. And it's specifically sensitive to thermal stimulation. Current treatments don't work well. And based on the data with NGF antibodies, there's a huge amount of data in patients with NGF antibodies. It's a validated target because the efficacy was very, very good. But NGF is a validated target for OA pain and also for chemotherapy-induced neuropathic pain. So we believe that ACD137, not having the side effect profile of the NGF antibody, is a very promising candidate for the treatment, both of acute and chronic neuropathic pain. So Pontus, I'll leave the word to you.
So hello everyone, my name is Pontus Forsell. I'm head of discovery and research here at Alzecure Pharma. Also one of the co-founders and preclinical project lead of the TrkA-NAM program. Today I will present a presentation entitled Next Generation Non-Opioid Analgesics. I will just give you a short background to the target pathway. So upon a release in the body, it could be as depicted here in the skin or in a deeper lying tissue like the knee joint. NGF is released from surrounding cells at the trauma site. This released NGF then travels to different kinds of cells where the receptor for NGF is expressed, and the receptor is called TrkA. So those cells could be immune cells like lymphocytes, macrophages, or resident mast cells in the skin. So there is a non-neuronal effect of NGF TrkA leading to increased inflammation at the site of the injury. NGF also binds to TrkA receptors on neuronal cells, as depicted here in this nerve ending in this figure here. So upon binding of NGF to TrkA, the nerve fibers will be sensitized. They will upregulate certain ion channels like TRPV1, NaV1.7, and NaV1.8, leading to allodynia at the site of injury and also a pain sensation. So this NGF TrkA pathway is very well validated. In man, it's actually validated by either deletion or mutations of the NGF or TrkA gene. These mutations or deletions lead to a painless phenotype. So these individuals do not sense pain. And that is, of course, not good because pain is the human body's defense. So you will not use the parts of the body that has been injured. And secondly, the pathway has also been validated by the anti-NGF antibodies, which actually demonstrated a step change in clinical efficacy in treating several types of pain. So to summarize this pathway, NGF TrkA pathway is very well validated both within nociceptive and neuropathic pain. So we'll talk a little bit more about the pathway and especially the neurotrophins, which is a family of protein hormones which NGF belongs to. So NGF stands for nerve growth factor, and it's one of the neurotrophins, as you can see here. There are other neurotrophins named BDNF or neurotrophin 4 and 5 and NT3, and they also come in pro forms. So we have a pro form of pro-NGF or pro-BDNF. And these different neurotrophins binds to different receptors. So I can see in this figure here, NGF binds to TrkA, and the other neutrophils bind to specific other receptors on the cell membrane. And the receptor to the right here is called P75NTR. It's a common receptor for all the neutrophils and all their proforms. So it doesn't only bind NGF, but it also binds NT4 and NT3. The different receptors have different functionality, of course, but they the TrkA, B, and C receptors signal in similar pathways. So that's probably spatial or temporal dependent. The P75 receptor has a totally different signaling pathway, more regulating cellular survival or apoptosis. So several attempts has been made to target this pathway for the treatment of pain. And the first attempt involved general inhibitors of both TrkA, TrkB, and TrkC. So these are called TrkA inhibitors. So they target the catalytic site of the receptor, which is a kinase domain. They are non-selective, so they inhibit all three different forms of the TrkA receptor, but not the P75 receptor. So these molecules were discontinued for the treatment of pain due to severe adverse events like CNS-related side effects. There are now two different compounds in clinical use for the treatment of different malignancies. So the second generation was the anti-NGF antibodies. So they are delivered by infusion, so not by a tablet or pill, and they neutralize NGF or pro-NGF, thereby inhibiting the binding of the ligand to the receptor. So the anti-NGF antibodies will inhibit both the P75 signaling as well as the TrkA signaling. So they are more selective than the TrkA inhibitors, but still the anti-NGF antibodies do inhibit two different pathways, the P75 receptor-mediated pathway and the TrkA mediated pathway. So the third attempt to target this pathway involves the negative allosteric modulators, of which ACD137 belongs to. So this is a more, and actually the most selective mechanism of targeting this NGF-TrkA pathway. These molecules only bind to TrkA, so they are highly selective towards TrkB and TrkC, and does not at all affect P75 signaling. And currently we are only aware of two different negative allosteric modulators of TrkA that is under development, and that is ACD137 and a compound that is in phase 2 right now for the treatment of over pain by Alzecure. So to summarize this, TrkA is a validated pathway for the treatment of pain. Allosteric modulators might give opportunity also for biased signaling because they are more modulating the activity rather than inhibiting it. This slide is just a summary showing different indications that the anti-NGF antibodies were being developed for. So these are 3 different antibodies, tanezumab, furlanimab, and fasinumab from Pfizer, Lilly, J&J, and Regeneron. They are either in phase 3 or in phase 2, and many of these studies showed a positive effect on the pain perception. So it gives a flavor of other potential indications that molecules targeting the NGF-TrkA pathway can be used in. And here we can see also different forms of peripheral or neuropathic pain, like diabetic peripheral neuropathy or cancer pain or post-herpetic neuralgia. However, these anti-NGF antibodies suffered some severe adverse events like RPOA, rapidly progressing OA, found in a small portion of patients, but still enough to be dose limiting for the anti-NGF antibodies. So why is that? Why did we see this effect of the anti-NGF antibodies? Well, our hypothesis is that it's due to the fact that the anti-NGF antibodies are not only inhibiting the NGF-TrkA pathway, but also the pro-NGF or NGF pathway mediated by P75, the P75 receptor. There are now quite many reports describing the involvement of P75 in bone mineralization or in bone homeostasis, suggesting that there is a link between P75 and potentially rapidly progressing OA also. It was also a nice paper in Nature a couple of years ago where they demonstrated that P75 actually limits the inflammation in the knee joint in an arthritis model in animals. So removing this brake by inhibiting the interaction between NGF and P75, which the anti-NGF antibodies do, will unleash that brake and you get an escalation of the inflammation. Also, the TrkA inhibitors that are on the market for oncology, like I mentioned, it's been given to a couple of thousand patients right now. It's larotrectinib and another molecule called entrectinib. These molecules have so far not been reported to have anything resembling RPOA as an adverse events. And finally, TrkA-NAMs have been demonstrated to have improved CNS safety profile. And in the bottom of the picture here, you can see an article or a figure from an article demonstrating what happens to the femur, which is lårben in svenska. And this is the cortical bone, so the dense outer layer of the bone. And if you knock out P75 from these animals, you can see that the density of the bone is much less than in wild types animals. And it also is similar for the trabecular bone, the more soft bone within the inside of the actual bone. So that sort of wraps up the background of this program. Now I will go a little bit into the actual TrkA-NAM program that they have here at Alzecure. So it's actually a program that goes back more than 10 years in the company. It was based on positive allosteric modulators of TrkA receptors that were identified early in 2013. So within this chemical series, we then, after a couple of years, identified selective negative allosteric modulators. So they were inhibiting the TrkA activity rather than increasing it as with the PAMs. Now, myself and our Director of Medicinal Chemistry, Gunnar Nordwall, we have worked on this target at previous positions in other companies, and we knew that identifying molecules with this kind of profile was not easy. So we decided to embark on a journey where we optimize this initial hit. And you can see in the figure to the right here that this initial hit is actually the black line to the right. And after lead optimization of this molecule, we were able to increase the potency of the molecules very much, and at the same time retaining a good selectivity towards other TrkA receptors. And AC137 was selected as a clinical candidate in 2024. So this is a slide showing just a summary of the TrkA-NAM program. Like I mentioned, we had a quite extensive lead optimization effort. We synthesized more than 860 molecules. We were able to identify or synthesize molecules with very good potency, below 10 picomolar, which is extremely potent molecules. In general, they have very good selectivity towards TrkB and TrkC, usually higher than 10,000-fold, and also solve the X-ray structure of the molecule. And importantly, they have, in general, low blood-brain barrier permeability, so meaning that the molecule will not be exposed to such a high levels in the brain, which is good from a CNS-related side effects. And we have finally demonstrated that several TrkA-NAM molecules have analgesic and anti-inflammatory effects in various in vivo models of pain. So why do we think that selectivity for TrkA over TrkB and TrkC is important? Well, All the TrkA receptors are involved in neuronal function, both during the development but also in the adult individuals. So it's important from a CNS side effects, but also from side effects related to peripheral nervous system that you are not targeting TrkB and TrkC. And this is a summary of different molecules, different TrkA-NAM molecules, also two TrkA inhibitors, as well as an anti-NF antibody here. TrkA-NAM is included in the table. And I just want to highlight ACD137 in the top row, where you can see we have an IC50 on TrkA receptor, approximately 1 nanomolar, and it's 20,000 nanomolar, more or less, on TrkB and TrkC. So given it's roughly 20,000-fold selective for the other TrkA receptors, and if you calculate the ratio for the other TrkA-NAM molecules, you will see that it's much less. And obviously for the TrkA inhibitor, there's no selectivity whatsoever between the different TrkA receptors. And interestingly, if you look at TrkA-NAM, which is extremely potent, it's 1 picomolar, the affinity at least for this antibody towards NGF, but it's not that selective actually. It's 1,000-fold selective towards TrkC, and there's been no reports on the selectivity towards TrkB. So actually the ACD137 molecule is the most selective molecule of all these different molecules targeting the TrkA-NAM or NGF-TrkA pathway. So I've been talking a little bit about BBB permeability and the importance of not targeting TrkB. So TrkB is intimately involved in memory performance or cognitive function. And here we have compared the TrkA inhibitor, larotrectinib, which is actually on the market now, in a model of memory in animals. And the higher the bar, the better the memory. And when we administer this TrkA inhibitor to these animals and then perform a memory test, we can see that there is a trend to decreased memory already at 3 mg per kg, and that's become significant at 30 mg per kg. On the other hand, when we administer the TrkA-NAM molecule, there is no effect of memory function, suggesting that we have, compared to TrkA inhibitors, at least a better side effect profile with respect to cognitive function. So summarizing this a little bit, selective and peripheral targeting of TrkA, we think that's a promising way forward for a new analgesics. We have identified highly potent negative allosteric modulators. We have synthesized, as far as we know, the most selective compound for TrkA over TrkB. And we think that this selectivity is really the key to obtain a safe compound with low side effects. But how about efficacy in animals? So we have tested it, like I mentioned, in several different models. This is an NGF-induced pain and inflammation model where we inject NGF. So it's a little bit artificial, but it's been used as our sort of screening models. So we inject NGF in the hind paw of the animal, and quite fast you will have an inflammation established within 10 to 15 minutes, and the animals will also have heat allodynia. And this is what we have tested to the left here, the analgesic effect of ACD137. You can see animals treated with only vehicle, they have a certain level of heat tolerance. This tolerance is decreased, so they withdraw their paw much faster when you have injected NGF, and you can see we can reverse this paw withdrawal latency in a nice dose-dependent manner with ACD137. And to the right, we also have the inflammatory effect of NGF. So you can see there is an increase on the inflammation. So that's actually the volume of the whole paw that we are measuring. And this we can reduce with ACD137. Going into a little bit, this is the inflammatory markers. Also, we have measured a couple of inflammatory markers through skin biopsies in these animals. And one marker that is really increased by NGF is CGRP. causing vasodilatation and thereby the resulting edema. This is increased a lot with NGF, and you can see we can block this increment with ACD137. And there is also a trend to an effect on IL-18 with NGF and ACD136. So we are not only reducing pain in this model, but we're also reducing biochemical markers of inflammation. So what about other disease-like models where pain is one component? This figure here shows the effects of ACD137 in a model of neuropathic pain, namely the chemotherapy-induced peripheral neuropathy, where pain is one component. So here we have used paclitaxel for inducing, which is a chemotherapy, and for inducing the neuropathy. And then we have administered ACD137 at 3 different doses and compared that to the effect of a known analgesics called gabapentin. And the higher the curve goes, the less pain the animals have. So here on the top we have naive animals, on the bottom we have in the black animals with peripheral neuropathy or painful peripheral neuropathy. And we can see at increasing doses that ACD137 can reduce this pain as much as 100 mg per kg of gabapentin, which is a quite high dose actually. On another model, this is a model of post-operative pain model, which is mostly a nociceptive pain model. So the animals have an incision, it's called the Brennan model, and then the pain is measured 24 hours after surgery. The same here, the higher the curve goes, the less pain the animals have. And you can see here that also there is a nice dose-dependent decrease of pain with ACD137 reaching, at least at 3 hours time point, reaching up to the same analgesic effect as tramadol, which is an opioid-based analgesics. So in these 2 models, both in a neuropathic pain model and in a nociceptive pain model, ACD137 gives a good analgesic effect. So we have also tested this analgesic effect in a model of OA, namely the MIA-induced model of arthritis in rats. So MIA or monoiodoacetate is injected into the knee joint of the animals, and after a couple of days there is an inflammation formed, and that will eventually lead to degeneration of the knee joint and a painful knee. So here we have administered ACD137 twice daily orally from day 3 to day 21 when we terminated the study. So here in this figure we have assessed pain by 2 different methods. Non-evoked pain, and that's measured on how much weight the animals put on each leg. And you can see here on the upper left of this figure is naive animals, and then they are given this monoiodoacetate, and that will change how much the weight they put on that particular knee or leg. So it's less weight. And here we can see that ACD137 is reducing that, so they distribute the weight more evenly. And interestingly, we have here compared the effects of ACD137 to tanezumab. It's a quite high dose given to these animals. And you can see that ACD137 is equally effective as the anti-NGF antibody tanezumab on non-evoked pain. And to the right, we have evoked pain. And you can see there's also a nice dose-dependent effect of ACD137 reaching up to the same analgesic effects as tanezumab, although it's a little bit hard to see. Tanezumab is the open circle on the top here. So essentially, both ACD137 and tanezumab more or less completely reduce the pain of these animals. So just wanted to say that this effect of ACD137 are as efficacious as the tanezumab anti-NGF antibody. But looking into a little bit more on the inflammation and also the joint pathology of these animals. So to the left, we have the inflammation of the knee joint. So it's measured actually by just measuring the diameter of the knee joint. So there's a quite pronounced swelling of the knee joint after administering this monoiodal acetate, as you can see here in the left part of the figure, and that is decreased in a dose-dependent manner with ACD137. And here tanezumab actually shows a little bit better anti-inflammatory effect with respect to the knee joint. We have also looked into the knee joint histology and tried to score the pathological events occurring in these knee joints. And you can see that the control animals here, of course, they have a perfectly healthy knee joint. Inducing arthritis in these animals will increase pathological condition in the knee joint, and that can be reduced significantly by ACD137. But this reduction was not statistically significant with tanezumab, maybe suggesting that targeting the NGF-TrkA pathway with a negative allosteric modulator could be more protective for the knee joint than targeting NGF. We looked a little bit more into that, and I will just show you this slide as a background to how the knee joint cellular morphology or anatomy of the knee joint. So this is a healthy knee joint to the left. The artery cartilage is built up of different layers. We have collagen fibers, both vertical and horizontal, and then different cells in different layers before the actual bone here. And to the right, we have a figure depicting what it looks like in a arthritic knee joint. You have swelling of the knee joint, less synovial fluid, and the cartilage will be affected. You will not have this smooth surface, but more a ruffled surface. The collagen fibers will be disoriented, and also the cellular layers are not so well defined. So there are several abnormalities here in this knee joint. And then we looked with hematoxylin and eosin staining of the knee joint of these animals that we had in the study. So this is just one figure from several, but wanted to at least give you a flavor of how it can look on a cellular level in the knee joint. So to the left, we have a healthy knee joint from naive animals. You can see the cartilage surface is really smooth. You have a well-defined cellular layer before the bone. Giving or administering MIA or monoiodal acetate to these animals will give a ruffled surface. You see, you will not have this well-defined cellular layer like we have in the healthy joint. And administering ACD137 seems to be protective. You have a smooth surface, only very, very small ruffles up here, well-defined cellular layers here, and that is not seen with tanezumab. So this it almost looks like non-treated MIA animals. You have ruffled surface, cellular abnormalities. So we think here that ACD137 or other molecules targeting TrkA in this same mechanism will have, at least in these models, have a protective effect on degenerative processes in the knee joint. So clearly an advantage over anti-NGF antibodies, I would say. And just to summarize this, we have identified ACD137 through an internally medicinal chemistry program, highly potent and selective negative allosteric modulators, similar analgesic efficacy as the anti-NGF antibodies, and ACD137 is currently in preclinical development. And with that, I thank you very much for your attention.
Thank you so much, Pontus. And we are now going over to a Q&A session. And Ludwig? please.
Yes, thank you so much for the presentation here. And as Martin mentioned, now we will now carry on with the Q&A. So if you have any questions, you can send them in via the form to the right. And the first question here is for Pontus. You have two pain products in your portfolio. What is the difference between TrkA-NAM ACD137 and ACD440 TRPV1?
Well, the most obvious is that they are targeting different proteins or targets within the body. TrkA-NAM is targeting TrkA, while ACD440 is targeting TRPV1 or BR1. So they are both proteins involved in pain sensation, but they are different mechanisms behind these two molecules. So that's the most important thing that distinguishes them. And then, of course, we have different ways of administering the molecule. The ACD440 is administered as a topical gel, while TrkA-NAM will be administered as a tablet or pill. So it's an oral administration. And they are suitable for different kinds of pain, I would say. So that's essentially what distinguishes them.
Thank you so much for that answer. The next question here is for Märta here. You observed both analgesic and anti-inflammatory effects with ACD137 in the preclinical model. What is the clinical relevance of the combination of these effects?
It's actually quite important because when patients have less inflammation, they have less swelling around the joints. So that means that you can actually mobilize your joint better. So that adds to your pain relief. And then having the pain relief on top of that increases function and quality of life. You can actually do more things actively and you will have less pain.
Thank you. The next question here is maybe both for Pontus and Martin here. I'll let you sort that out. In the MIA model, ACD137 had protective effects on the knee joint. What does this mean for the project from a scientific and commercial standpoint?
Let's begin with the science.
Okay. Well, so from a scientific point of view, we think that the effects that we have seen suggest that there could be a disease-modifying effects actually of the molecule, or at least a knee joint protective effect, which is not seen with other kind of molecules like the anti-NGF antimolecules, which actually have this, if anything, the opposite effects. Well, they induce rapidly progressing away, although in a very small set of patients. But I think that's really a key feature, and also the anti-inflammatory effects of the molecule in this model is of importance.
And if we go over on the commercial side or perspective of this, I would say if we would only have a symptomatic effect and pain relief, let's say that, and I think this is a fair prognosis regarding sales. Sales would potentially be in peak sales around 1 to 2.5 to 3 billion. Looking at forecasts from experts within this field, when you have a so-called disease-modifying and protective effect, these forecasts say that sales could be expected to be doubled, so up towards $5 billion in peak sales. And of course, this makes this program even more interesting and attractive from a commercial perspective.
Thank you so much. The next question here, how would you expect to position ACD137 to all OA patients or a subcategory?
Märta, please.
I would actually say that it would be applicable to the whole group of OA patients because of its both anti-inflammatory and pain-relieving effects. So I would not see necessarily that there is a need for a subgroup that would be responders. I mean, you could always say that, okay, if you don't tolerate, you could think of it as a second line, but the features we have and the safety that we seem to have so far, we're looking at safety studies, of course, as a next step. It actually should outperform everything that's on the market now for the whole group.
And I think to Märta's point, it's all about efficacy. If you have a better efficacy, you would be chosen as a first line. And with the alternatives that are on the market today, they are not sufficient, which would position TrkA-NAM in a very positive position.
Yeah, and just adding on to the safety, because what actually makes people stop taking treatments and make the doctors stop prescribing is actually a lot of safety rather than lack of efficacy.
Thank you. What endpoints do you expect in proof of concept and pivotal clinical trials? Are there any good biomarkers one can use?
Well, the biomarkers for the OA pain, I would say that the market or the arena for OA treatment has been, there's very many drugs that are approved for the indication. So that means that the outcome measures are very standardized. There's something called a WOMAC scale. So you actually measure morning stiffness, how painful it is to walk the stairs, if you have pain at night and things like that. And of course, you can always measure always swelling, but in order to find, to have an efficacy that's good enough, I don't think the biomarkers are that important actually, but it's something that we of course need to look into.
Thank you.
There are anti-inflammatory biomarkers, of course, and there's drugs, potential competitors that are looking purely on the anti-inflammatory side, and they could have some effect on the anti-inflammatory part. But I believe that the pain relief and also the lack of safety problems when it comes to joint destruction are the key features.
Thank you. Do you expect ACD137 to have a disease-modifying effect in OA?
Okay.
I mean, given the findings in the animal study in the arthritis model, it's not unlikely, although we of course don't know until we have done the clinical studies, but the combination of an anti-inflammatory effect, of course with an good analgesic effect, but the combination of an anti-inflammatory effect with this protective effect, which we saw in the knee joint, could suggest that there is a disease-modifying effect also in OA in man, but future will tell that definitely in clinical trials, I would say.
Thank you so much. And Märta, maybe you could tell us a little bit, how does the competitive landscape look like?
Well, apart from all the drugs that are on the market, really, so non-steroidals of different types, there's a number of products that are in the pipeline, so to say, so are in clinical development. I know there's some TrkA inhibitors and NAMs on the market or in early development, nothing really established yet. But when you look at phase 2, phase 3 programs, these are basically anti-inflammatory antibodies or proteins, injectables. So there's not really that much that is just specifically toward pain. I'm sure that there's nothing that's directly versus the pain mechanisms per se, but I would say there's 3 products that are right now in clinical development, but they're all also injectables. So of course it's easier with a pill a day. That's where we land.
Thank you so much. And Martin, this is still an early asset. What is the interest from the market here?
We see active interest in TrkA-NAM, and we have seen it increasing. So it feels very positive.
Thank you so much for that. And maybe one for you here, Pontus. Why would a TrkA-NAM molecule be safe when both anti-NGF antibodies and TrkA inhibitors have failed due to safety?
Well, I would say that the more selective mechanism of ACD137, not targeting TrkB or TrkC or P75 NTR, is really key here. And this is also supported, that it's a safe mechanism. It's supported by the finding with the protective effect in the knee joint. So I would say that those 2 things are really beneficial for the ACD137 molecule and TrkA-NAMs in general.
Thank you so much. Another question we have been sent in to us here. Paxman develops a product for CIPN that cools the hands, thereby limiting chemotherapy's exposure to the nerves. What is your view on this?
I think that would be fantastic. We haven't really seen the results yet. We've seen we know that the trial is there, and we know it has completed the clinical phase, but so far we're not aware of any results that have been published.
Thank you. And another question for you, Märta, here as well. Is TrkA-NAM a molecule more or less effective than a NAV 1.7 or NAV 1.8 inhibitor?
Well, if you look at NAV 1.7 or NAV 1.8 as oral or injectables, that's one thing. If you look at NAV 1.7 as a topical, that's basically a lidocaine that you use for insect bites, etc. So it's not taken up. But what they do, these two NAV channels do, is that they affect how the pain signal is transmitted through the nerve fiber and up to the brain so that we can perceive pain. The TrkA-NAM molecule actually reduces the irritation or the start of the signal in the nerve fiber. So I would say that if there's nothing starting the signal, it's much more likely to have a broader effect than a NAV 1.7, NAV 1.8, because the input to the nerve fiber is actually what drives the signal at the very end. Of course, there is a NAV 1.8 inhibitor that has been approved now for postoperative pain, and it's about as efficacious as a Tylenol or very weak opioids. So I think that for chronic pain, and I think the TrkA-NAM is a much more likely probability of success going forward. Right now, they're looking at NAV 1.8 as an oral product for chronic pain, and so far we haven't seen the results very promising results from that target. It would be great to have options, and especially if you could combine them, but we're not there yet.
Thank you so much for that. What is the timeline for ACD137 to come to a clinical trial? What preclinical work is left?
We are preparing for safety talks, so that's where we are, and that is what we are preparing for. But specific timelines we don't want to give.
Thank you for that, Martin. And Pontus, why is a small molecule preferable to anti-NGF antibodies?
Well, I would like to come back to the selective mechanism, of course, not targeting the other neurotrophin receptors, TrkB or TrkC or P75, but also in general, of course, the ease of use, a pill versus intravenous infusion. It's much easier for the patients. We also have the cost of goods, where a small molecule often is cheaper to develop and synthesize than large molecules. So I think those two really gives a small molecule the upper hand.
Thank you so much for that. Moving on to the last question here is one for you, Martin. Do you plan to take TrkA-NAM project into clinical studies or out-license the program?
We have begun to develop the plans for clinical studies. We are having dialogues with regard to out-licensing, and if we will out-license or not depends on who we are talking about, the offers we are proposed, as well as our internal our alternatives with regard to our cash position and out-licensing of other projects. We know that TrkA-NAM is a very attractive program, and the longer we can drive it, the higher value we can develop. And of course, that would be ideal for current shareholders. So we are optimistic and are keeping all options open.
Thank you so much for that. As I mentioned earlier, that was all the questions we had for today. So I will now hand over the word to you, Martin, for some concluding remarks.
Okay, thank you so much. So let's go to take-home messages based on today's presentations. And what we can say is that there is a very large unmet medical need, both in osteoarthritis as well as in chemotherapy-induced neuropathic pain. And these patient groups are growing with the aging population as well as overweight problems. Today, more than 1 billion individuals are, for instance, suffering from OI, and sales last year forecasted to have hit 10 billion and expected to grow 5 to 8 in the coming per year in the coming 10 years. So a huge market and opportunity. And as we have heard today, we have a strong validation for TrkA-NAM that NGF-TrkA pain signaling is an attractive route to go. And we have data here, which is both genetic, preclinical, as well as clinical. And ACD137, our TrkA-NAM, is a negative allosteric modulator blocking on NGF signaling. And we have several attractive molecules where ACD137 is our lead candidate. And with a more selective mechanism, as we have heard today, we hope to be able to offer a safer treatment than anti-NGF antibodies, which showed a very attractive efficacy. And as we heard here today, TrkA-NAM under Alzecure is under development as an oral medication, which we think is key when you want to go in and treat large patient populations. And we are preparing it for clinical trials. So with that, I would like to thank everybody for listening in today. I recommend you to download our recent article showing new results comparing with anti-NGF antibodies. And Ludwig, I'm handing over to you.
Thank you so much for that, Martin. And yes, well, thank all of you for tuning in and sending questions. And I wish you a pleasant day. Thank you so much.